Welcome back. Second half of the action potentials lecture. Some examples and tangents and consequences. So tangent number one, how do we study all of that? How can we study action potentials? How can we study what neurons do? Right now, um, I just realized that on this picture, I actually don't show the historical experiments with axons from a squid, but one technique that people do these days is connecting to neurons with special electrodes, with glass electrodes typically. So you use an electrode to connect to the inside of a cell and record electrical currents in the cell. And all of these pictures show, it's just some random pictures from the internet, show electrodes, so you can see them as these strange shapes, but actually these strange shapes are very tips of glass pipettes, of glass electrodes filled with conductive fluid that people use in order to connect to cells and kind of listen to them in the electrical cells. So detect electrical currents in the cell. And that's a very powerful technique, and we'll talk a little bit about it in the future. All right, another important tangent. So we discussed how the rising phase of the um, action potential, when the uh, positive feedback happens, is like this explosion thing where you have a little bit of sodium entering the cell that depolarizes the cell, that opens more channels, that lets more sodium enter. But what starts the process, right? Because we described how the process can self-perpetuate and amplify, but there has to be this initial depolarization to open the first channel, right? The way we described it right now, we don't have, so this is something that so far we haven't mentioned. What starts the process? In case of an explosion, typically, if everything goes right, when people use explosions to build things, for example, to build um, roads through the mountains, somebody needs to send this initial signal that will, like a spark or something, that will actually make things explode. So what is the event that triggers action potentials? Okay, that's a good question actually. So pause the video and think, what can be the reason for this initial opening of a few channels? We don't have to open a lot, but we need to open a few channels. We need to depolarize the cell a little bit in order for the process to start running. So what can be the thing that causes this initial depolarization? Think about it. Pause the video, think about it. Yep. I will just show four answers, four possible answers. So there are at least four answers that I know to this question. And the first answer, for which we don't have an image, sometimes it just happens. So most neurons, or at least many neurons, are capable of just spiking spontaneously. And maybe they don't spike spontaneously, and spontaneously means out of nowhere, right? They just do. So sometimes they don't spike spontaneously in norm, but if they're silent for too long, then they kind of generate an action potential. But some neurons are just going like that. So some neurons have lots of noisy spikes, and they just keep producing these spikes no matter what. So, yep. Sometimes it's periodical, sometimes it's just because of the noise, sort of, that's built in the system. So their voltage fluctuates, and every now and then, like, pew, you have a spike, pew, you have another, then no spikes for a while, then pew, yet another spike. That's one option, and that's a legit option that's actually, we will even discuss next week. Yeah. Next week we will discuss how sometimes how this spontaneity of, like, spontaneous generation of action potentials, how sometimes it plays a crucial role in animal survival. But then there are also three cases that are kind of more mechanistic in the sense that, okay, something happens, something else happens, and it just triggers an action potential. So first of all, sensation is one of those cases. So maybe the neuron is a sensory neuron and it's connected to something, or it senses stuff on itself. And actually, we just a few minutes ago, we talked about stretch receptors, remember? And neurons that we use for touch. So we may have a neuron that has voltage gated channels, but also has stretch activated channels. And when you touch something, at first you open a stretch activated channel, some sodium enters the cell depolarizes, and then it spikes, and the spikes travels from, say, from your fingers to your brain, because actually that's what happens. Neurons that are sensitive in your fingers, they have axons that go all the way to your brain, which is amazing. Yeah, that's one option. As probably more common option is that this neuron sits somewhere in the middle of a network, and so other neurons talk to it. And when they talk, when one neuron talks to another neuron, there is a thing called synapses or synapses that allow one neuron to excite another neuron. So one neuron spikes and then it like talks to another neuron, and then the other neuron spikes. And we will discuss that in 2 weeks from now. And we will talk about it in detail, and we will talk about how synapses work. But yeah, for now, it's important to know that neurons can excite one each other. And finally, the most common and almost like trivial reason is that when a neuron is long, when a neuron has an axon that is long, then action potential in this axon doesn't happen instantaneously at all parts of it, right? The whole point is that it propagates down the axon. So when you touch something and an action potential starts, it actually runs through an axon that goes through your body, and it takes a little bit of time for the signal to reach from your hand or your food to your brain, which actually will even be important in some cases. Say if you step on a thorn, like on this water chestnut seed that we have so many around here. So that a spiky, if you step on a thorn, you really want to move real quick before the damage is done to your food, and the delay time will become quite of an essence. So, and delay time, the propagation delay, will become noticeable at this point, right? And so when the signal propagates through an axon, it's like a forest fire in the sense that an action potential in one part of an axon causes an action potential nearby, and then nearby, and then nearby, and that's how it propagates. And we'll return to that in a few seconds, in a few minutes. Okay, good. So that's how everything starts before the positive feedback kicks in. Now, here's a little exercise to just drive the point down. So imagine that some scientists caught a neuron and they connected to this neuron with electrodes, and now they can inject currents into this neuron. And that's a fancy terminology. People actually use this terminology. They inject currents, electric currents. You would say about injecting, you would talk about injecting currents in the same way as people talk about injecting chemicals. But what they actually do, they just send some electricity into the neuron, and they can bring the neuron down, so they can try to hyperpolarize neuron to decrease its potential with electricity, and they can try to increase its potential. And so here's a picture from some textbook that I kind of like, and that's why I copied it here. So imagine that first they recorded, like first they did nothing, this is nothing, and then they tried to um, decrease the potential a little bit, and then a little bit more, and then they try to increase the potential a little bit, and then a little bit more, and yet more, and yet more. So if you record the current, no, let's say better, if you record the voltage, the membrane voltage, so how the neuron responds to these attempts to change its potential, what will you see? How will the voltage change over time? Here, and like here, and like here. Okay? This is the question. make your bets, pause the video, ideally take a piece of paper, draw it, and then I'll show you the answer. The answer, here's what happens. If you try to hyperpolarize the neuron, so you make the potential even lower, well, the potential just goes down, but the neuron is already hyperpolarized, so it doesn't spike, and you just move it further away from the threshold, from the point at which the positive feedback kicks in. And so in most neurons, it just will it will hyperpolarize, and then when you release it, it will come back and nothing will happen. There are special neurons, like you can actually, biology is fancy, so there would be neurons that would respond to that in some weird ways, but for a typical neuron, like for the most common run-of-the-mill neuron, yeah, it will hyperpolarize and then it will come back to the equilibrium resting membrane potential, like, yep. No consequences. Now, if you depolarize it a little bit, if you depolarize it a little bit, like here, I drew something giant. Okay, if you depolarize it a little bit, then it will get depolarized a little bit, but until a certain point, nothing will grow out of it because there is a certain threshold level, right? It's shown here. So we don't know it a priori. I mean, for every neuron it will be different, but up to a certain point, voltage-gated channels won't open, because they definitely don't open at resting potential, and they definitely do open when the neuron is depolarized. So there has to be some threshold where they go from the closed state to the open state when they start opening. And until you reach it, nothing changes. But once you reach it, then here, see what's happening. So first we have the passive depolarization, so this part, and then we reach the threshold, and then we immediately shift to entirely different story, because now, so until then, it's just the current that the scientist injects into the neuron. It gradually changes its potential, but then at this point, okay, the first channel's open. Sodium starts to get in, which increases the potential even more, and so you have this change from a slow passive process to a fast process, and you have this explosion of potential. It goes up real quick, and then later channels inactivate, potassium channels open, and it goes down, and you have an action potential, right? In a real brain, it won't be electrodes connected to the neuron, but it would be things like inputs from other neurons, synapses, as we talked just in the previous slide, and as we will talk in 2 weeks a lot. But now the question is, what will happen if we inject even more current, if the neuron is activated stronger, if more synapses get activated? Well, here's an interesting thing. The action potential, the shape of one action potential actually doesn't change. There are no small action potentials and large action potentials. Action potentials are always the same, because if it's exploding, it's exploding. It's like with explosions. You cannot have a tiny explosion if you have always the same amount of stuff. Like if you have a hunting gun, you cannot do like a little, like fire it a little bit and then fire it more as long as you have the same sort of ammunition. Like, I don't know much about hunting guns, but I imagine there is ammunition that you put in a gun or something. So if you have a process like that, it always happens in the same way, right? So what can change is the number of events. Like if you're fighting, if you're hunting, what do people hunt? I don't like it, this example. Zombies. Okay, zombies. If you're hunting zombies and there are lots of zombies, then yeah, sure, you fire lots of shots, right? Because there is a crowd of zombies. But if there is only one zombie, then you're like casually like fire a warning shot and zombie goes back to the zombie land and just sits in the swamp. So you can change the number of events, but the events themselves don't change, because regardless of what happens, it's still like as much sodium will enter as it can before it inactivates. I should use this hand. Before it inactivates, and then potassium opens, potassium channels open and bring everything back to the initial state. So as we here on this picture, as we inject more currents, what happens is that a stronger current, what happens is that we have more action potentials, but the size, the shape of each individual action potential doesn't change. They just are packed closer to each other. And there is a limit to, like, they cannot be closer to each other than something, because it takes some time for the thing to recharge, like go up, go down, recover, because sodium channels should stop being inactivated and potassium channels should close, and it takes a little while. So there is this refractory period thing, the smallest period, that the smallest amount of time that needs to happen between two consecutive action potentials. And for some neurons it's longer, for some neurons it's shorter. So some neurons fire a lot at high frequency, and some neurons are like, nope, I'm only doing it like once every 10 minutes or so. Everything is really variable, but yeah, the refractory period is always there. Good. Next, are all sodium and potassium channels the same? I guess I should have asked, are all voltage gated sodium and potassium channels the same? What do you think? Of course not, right? There are different neurons. So what's different about different voltage gated channels? Why are they different? What makes them different? Well, so channels are proteins, right? Where do proteins come from? They are synthesized by the cell based on the records, the genes in DNA. So we have like DNA, and then I should have prepared that, and then something, we have protein. I don't like writing. Okay, protein. What happens in the middle is actually RNA is involved, and we're not going into that right now, but actually DNA is copied to RNA. RNA is sometimes edited a little bit, then goes to a special place where it's read, and the protein is combined, and then protein assembles. At first it's combined as just a sequence of amino acids, and then like it assembles into functional thingy, into functional machine, because most proteins are machines. Some of them are structural, so some of them are like more support structures. But anyways, what's important is that we have genes, and out of genes, out of these records in the DNA, proteins are synthesized, which means that we can have several records, and then we'll have several proteins that are close to each other, that are similar to each other, but still slightly different. So we can have several different types of voltage-gated sodium channels and several different types of voltage-gated potassium channels. And indeed, that's the case. We can also modify them a little bit. So, and by we, I mean like we, the living things. There are different processes in the cell that can fine-tune kind of the state of each individual protein. But because of that, the kind of result of all that, of the diversity of voltage gated channels, is that we have diversity of spiking. So different cells spike really differently. Some spikes are wider, some are narrower, some can spike a lot, some spike very little. Then, as you can see on these pictures, some cells kind of spike really periodically, so they can do this bum 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 thing that sometimes can be really helpful for some processes. We will talk about that. And some cells, they spike at a higher frequency in the beginning, and then the frequency goes down later. And it also can actually be a feature and not a bug. It can be utilized by the system as a whole. It may play a role in how brains work and how information is processed. And then some cells can be in this bistable situation, when sometimes they are silent and sometimes they're bursting. So bursting cells, bistable cells that can like silent, then really active, then really silent, then really active. There are all sorts and kinds of cells, and people who study that, you can go in really in depth with studying why it is so, and what are the mechanisms, and what are the consequences when something goes slightly wrong, and how to fix it, and all the interesting questions that one can ask about that. Next, can any chemical prevent channels from functioning? Sodium and potassium voltage gated channels. What do you think? Can there be a chemical that would break this channel somehow? The answer is generally yes to a question like that. Yes, there may be a chemical that would break them. And as we talked before, you know, we mentioned one thing like that. So chemicals that break something are usually like poisons and venoms, right? And so there is this funny fish, not the big one, but this like round shapy fish. This round shapy fish over there, it's called the fugu fish, and it's very popular in Japan. Have you heard about it? It's also popular among tourists who like it's interesting. If you haven't heard about it, read about it, but most people have heard about it. So it's very poisonous, and if you eat it, you die. But the poison is actually not in the flesh, not in the meat, but in some other structures in the body. So in theory, it's possible to cook it, to carve it in such a way that the meat gets separated from the poison. And so there are special cooks that are specially certified that can cook it. And for some reason, people keep eating it fish because allegedly it's very tasty. But every now and then, like almost every year, somebody dies because there are some people who pretend to be cooks and have fake certificates on some islands and somewhere, and they try to basically, bootleg chefs, and people try to save money and die because the toxin that this fish has is called tetrodotoxin, and it's pretty famous among scientists. It blocks sodium channels, voltage gated sodium channels. And the reason it's famous with neuroscientists in general really like toxins and venoms and toxic plants and stuff, because evolution came up with all these wonderful weapons that allows one animal or plant kill other animals or plants, or at least make sure that they never eat them again. And so the evolution took care of finding lots and lots of interesting chemicals that are really helpful in research, because now if you have access to this chemical, you can block all sodium channels, and you can just, you can probably imagine that in many situations it may be handy to be able to selectively block only one type of channels but leave other channels functioning. All sorts of interesting questions can be asked. And so tetrodotoxin blocks sodium channels. Now, let's see. What's your guess? How would people die if they eat this fish in a like without cooking it in this special elaborate way? What will happen if you block all sodium channels? Let's actually like think about that. What will happen to a living being if you block all sodium channels? Well, if you block all sodium channels, there are no action potentials. So at the very least, there is no thinking, right? No information processing. Will the living being actually die? Yeah. Why? What will kill it? So no action potentials in neurons. Usually when I ask this question, some people say the heart will stop beating. Actually, in mammals, the heart won't stop beating because the heart doesn't rely on neurons to beat. It has action potentials, but it is a muscle. Like heart is made of special muscles, and action potentials in these muscles are that's what makes the heart beat, and tetrodotoxin doesn't affect that. So the heart continues beating. What other thing, when it's off, kills an animal? Breathing, right? So the animal will stop breathing, and they will die. And actually, in case of tetrodotoxin, everything is slightly weirder because the action potentials in the head don't stop. And the reason for that is a special thing called blood-brain barrier. So most things in our blood don't automatically get to our brain because there is a special thing, a special fancy interface between the blood and the brain. So and it's a problem in pharmacology, for example, because many drugs, many pills, many special substances that would have helped actually don't cross blood-brain barrier. So you cannot just give a person something if it would have affected the brain positively, but it doesn't get to the brain, because what happens there is that brain cells don't bath in the fluid that's like in direct contact with blood, but then there is a special, really careful selection of molecules. So glucose passes, oxygen passes, some small molecules like ethanol, like alcohol. It's so small that it actually passes. But for most molecules, they are actively transported into the brain. So that's a fancy topic. We'll talk about it later, probably. But it's relevant here in this picturesque and gruesome way, because if somebody eats these toxin, they actually are fully conscious, but they get paralyzed, and eventually they die of asphyxiation because they cannot breathe. And you can save these people if you put them on a respirator for about a week or two, I guess. I think, if I remember it correctly, because it takes that much time for the um, toxin to be eliminated. I think, yeah. So, right, but the problem is that if a person is on an island and tries to get this thing for cheap, then probably they're far from a respirator. So people die because there is not enough time to bring them to a hospital. Yep. All right, so this was about blocking the sodium channels. And in this case, it's a toxin produced by fish. Oh yeah, here's the fish. I should have showed it before. Here's the fish. Oh, by the way, it reminds me that another reason. So why do people keep eating this fish? Allegedly, one reason is that it's tasty. I cannot imagine how a fish should be so tasty that people would risk their lives. But whatever. Maybe some people care about their food that much. Another reason that probably people like the thrill, like, oh, I can die, and it's probably expensive, but I can die and it's expensive, but I can still afford that. I guess it's an adventure of sorts. But allegedly, another reason is that it makes people feel a little bit funny because some residual amount of toxin is still there and it still has some effect on the brain, apparently, even though supposedly it doesn't cross the blood-brain barrier. So I don't quite understand that, but there are indications that people feel a little bit differently. And also, there is an interesting story that apparently dolphins get high on this fish because they like take this fish, and these fish are not normally puffed. They puff in order to scare people and like predators. So dolphins get this fish and then poke them with their nose, and fish releases a little bit of this toxin in the water, and dolphins get high on it. Apparently, I don't know whether it's true, but the behavior is described. Maybe they just look funny, these fish. I am not sure. You can look it up. Okay, next. So that was the tetrodotoxin was about inactivating channels. Can you activate channels? Like, what will happen if you activate channels? If you have another toxin that, instead of closing channels, instead of blocking them, opens them by force, what will happen if all sodium channels are open, get open in a body of an animal, like a fish? What will happen if all sodium channels are open? Think about it. Make your bet. It's not an obvious, it's actually harder to imagine a little bit than with blocking. So make your bet. Okay, all sodium channels are activated. What happens? First of all, action potentials cannot happen, right? Everything is depolarized, but it cannot hyperpolarize back. And so it's like one-way road now. It hyperpolarizes and stays like that. And everything is active at the same time. So it's kind of like paralysis as well, except it's not a paralysis when nothing works, but it's a paralysis when everything is engaged and stays like that. And also, sensory neurons use spikes, right? Sensory neurons that sense information and send it to the brain, they also use action potentials. And in this case, all of these neurons are engaged, including pain neurons. So it's incredibly painful. And these pictures on the background are pictures of cone snails. And cone snails are horrible, vicious predators. They crawl. I mean, the shells look so awesome, but they actually they go on the surface of, I mean, on the bottom of the ocean, and they have this special tentacle, and they can inject venom into their prey, and then they eat it. And here we have a picture of it injecting venom in a fish that's now dead or dying, and they can bite humans, and apparently it's incredibly painful and dangerous in some cases because these snails produce a whole concoction of um, I'm not sure I'm saying this word right. I shouldn't repeat it. So, but um, lots of different toxins, and some of them, all of them, are called conotoxins because they're produced by cone snails. But there is no like all of them affect different channels, but some of them affect sodium channels. And here above, you can see people actually growing cone snails and collecting their venom. And that's actually what people do because they produce so many amazing substances that then later are separated and sold for like $100 a vial for researchers because they are selective. Some of them are selective blockers or activators of different channels, and some of them activate sodium channels. Very painful, also deadly. Okay, next question. In our human life, are there cases when we want to block sodium channels? So, I mean, fish is weird, right? So, barring the eating of the weird fish, are there cases when we may want to block sodium channels? When? Why? What's the use? What's the point? Maybe not in the entire body, huh? Maybe only in part of the body. Would it help? Think about that. Did you get it? The answer? Say surgery. And I mean not anesthesia actually, not like global anesthesia, not a big surgery because it's too risky to try to inactivate sodium channels in the entire brain because the person will probably die because just switching off the brain is not a good idea. But if you have a local surgery, then you can use thing like a lidocaine, or maybe like if you have a hole in your tooth and a filling needs to be made. So people made these injections and injections of some sort of anesthetic, and all these anesthetics, local topical anesthetics and local anesthetics that get injected in the nerve or near the nerve, they block sodium channels because then the action potential will start going, but once it reaches the part where sodium channels are turned off, it cannot go any further, and so the um, dentist can like fix your teeth without you um, suffering. And just a few years ago, essentially just a few decades ago, it wasn't possible, and it was like, fixing teeth was a much more painful and much more dramatic affair. So thank you, lidocaine and other anesthetics for making small surgeries and medical procedures possible. Next, what about natural pain regulation? So in this case, we use a chemical to change the ability of neurons to transmit information to generate action potentials, and it changes our pain perception, pain processing. It makes certain things numb and painless. Do our body have the same ability? Do we change how painful different objects are, different things, different events are? And I don't mean like metaphysical pain, I mean like real pain when something hurts. Do things always hurt the same way, or can it be the situation when pain is less noticeable or more noticeable? So let's think about that. But before we think about that, like why pain is even a thing? Why do we have pain? And there is a reading about that below, but also like I should show it below, but also let's just, even if you haven't read it, let's just think from common principles. So why do we have pain? Pain is unpleasant. Why do we have it? When things hurt, why things hurt, and what would happen if we wouldn't feel pain? Think about that. Okay, what will happen if we don't feel pain? It turns out that we know what will happen because there are medical conditions when people don't feel pain. And one condition that's sad, all of these conditions are sad, but one sad condition is leprosy. And in leprosy, people gradually lose fingers. And yeah, I'm sorry for showing a picture that's kind of painful, but people lose fingers, and for a while, it was assumed that leprosy just eats fingers away, sort of, that people just, that, they disappear. But it turns out that no, that's not the case. It just leprosy, when people have it, it kills, it destroys pain fibers. And so people stop feeling pain. And if you live in a country where you have to do stuff with your hands all the time, when you tend after a fire, when you have to chop wood, when you have to work with sharp objects all the time in your household, what happens is that you hurt yourself all the time. And gradually, as people hurt themselves, they don't notice that and they continue hurting themselves, and they lose their fingers and their proximities, despite the fact that the disease actually doesn't cause it, it just removes pain. Because pain uh, communicates to us that something is wrong and something should be taken care of, right? And there is this reading in the, like, in the information uh, section accompanying this video that I recommend that you read, about cases when people don't feel pain from birth. But so in a normal human, like normal is a charged word, but in a typical human, what's are there cases when pain becomes more and less noticeable? Like, let's come with a case when pain becomes less noticeable, like a real thing that happens to real people, and a situation when pain becomes more noticeable, and maybe even things that are not normally painful become painful. Like, why, and why is it helpful? Think about that. One case when pain becomes less noticeable, when things that are normally painful stop being painful, and one situation when the opposite is true and things that are normally not painful become painful. Come up with 2 examples, please. Okay, so when painful things are not painful, for example, when a tiger is chasing you, and if you have a cut, it would be better for you if you don't pay attention to it, right? So if you're fighting a tiger or running away from a tiger, but especially fighting, you may have some damage, you may get some damage, but at first it won't hurt. And it's called shock, it's called intense stress. But actually, it's not about sodium channels. So this change in pain perception actually happens in the central nervous system. And we'll talk about it later. It's not about sodium channels directly changing the way they open and close. So it's not like analgesia, that like this Novocaine and stuff that people use as a local analgesic. The opposite, however, when normally non-painful things become painful, are related to sodium channels changing their function. And I really like this stock image, because the person hurts, but also it's obviously fake, so it's not gross. And the hand is red, but that's actually Photoshop. But in real life, when you hurt your hand, sometimes things that are normally non-painful, like touch, become painful. Why? What's the point? I mean, why? There are at least 2 types of why in biology. Why in terms of, like, why evolution selected for that, why it became a thing, in which way it helps to an animal, a person, a living being, that it justifies the existence of this feature, of this fancy feature. That's one kind of why. And another why is more like how does it happen? What is the mechanism for that? So why, in terms of evolution, why it's good when your hand hurts more than usual because you cut it or burned it? What do you think? Well, if you burned it, if you cut it, you'd rather care about it, right? And pain makes you care about it because now it's tender and vulnerable, and so it hurts to even touch it. So you tend to put it in such a way that you don't touch it, and the chances that it recovers increase. So you don't like use it, your daily routine. You do everything with your other hand, something. So that's why it makes sense. How does it work? Well, when you have inflammation, there are special chemicals that are released by the inflamed tissue, and among other things, they do lots of things. They make everything red and swollen because they change how blood works, but among other things, they affect how pain sensitive neurons spike, and it becomes easier for them to spike, and they even start spiking to things that they don't usually respond to, like touch or heat or cold maybe, but heat is, and touch especially. And so mild heat that's normally is just warmth becomes painful, and same with mild touch. And that's because of modulation of sodium channels. Next question: what happens if a gene for a sodium channels mutate? For a sodium channel, for a voltage gated sodium channel mutate? Um, and that's an involved question, but yeah, I guess still let's try. So what happens if a channel becomes, sodium channels become less eager to get activated or more eager to get activated as a result of a mutation? So is there from the birth of as the human, a new human is born, and in the genome there was a tiny mutation that appeared, and so this gene is slightly different, and the protein is slightly different, and the protein is slightly dysfunctional, and this sodium voltage gated channel doesn't work as it's supposed to. Either it opens too eagerly, or it opens, or it doesn't want to open. What changes? What do you think? Make your bets. Did you make a bet? Let's compare to the answers. And the answers, this is actually a table from Wikipedia that I really like. And from this table, we can learn like 4 things or something. So first, Wikipedia is your friend. Wikipedia for channels and for genes and for stuff like that is actually pretty well written. In general, with Wikipedia for niche topics, it's more likely that the information it contains will be weird. But for topics that many people care about, like these medical conditions, for example, it's usually okay as a starting point. It's usually a pretty decent starting point. And so that's learning point number 1. Learning point number 2, as you can see, there are lots of different types of voltage gated channels. And they have names, as you can see. So they're all like Na, which is sodium, V, which is like voltage gated, and then numbers. So yeah, there are all these subtypes of channels, all these different genes, channels corresponding to different genes that we talked about. And if you look at the rows, okay, some of them are more in the central neurons, but some of them are more in the muscle, and some of them are more in the cardiac muscle, cardiac myocytes, and some of them are more in the like neurons that are in the body and stuff like that. So different channels are used by different cells in different tissues, perhaps. That's learning point number, I guess, 2. Learning point number 3 is that for each protein, we have a gene, and proteins and genes have slightly different names. And a table like that is really important if you want to learn more about something like that, because now what you can do, you can take this name of a gene and put it into a search engine and look for all papers, all scientific papers that mention it. And I will have a separate video about how to do that and how to find stuff and how to use things like Google Scholar. But the main idea is that now, once you have this code, you can find information about this gene in particular, and all the case descriptions and all the stories and all the studies that try to understand what this gene actually does. Right? And now finally, let's look at functions of voltage gated channels. How can we figure out the function? One of the ways is to look at channelopathies. So to look at disorders and problems that people have when this gene mutates just a little bit. If it mutates a lot, it's actually so sad that probably with these genes, if it's completely absent, most probably the animal, the person, the embryo cannot be alive, because these are all pretty fundamental and involved genes. So you cannot just forget how to spike. If spiking is gone, nothing works to such an extent that probably the embryo won't develop. But if the change is subtle, then we may be in this space where the animal or a human, an animal, if it's well, animals also have diseases, right? Or maybe it's a model animal that people use to study how to fix this disease, or in a human, in a patient. So if it's just slightly off, then the person will be alive, but there will be consequences. And so what can be the consequences? If you look at that, there are lots of different words here, but one word that's pretty consistent is epilepsy, and look, spasm, and another epilepsy, seizures, epilepsy, and more of that, I guess, later. So if sodium channels work, voltage-gated sodium channels work not how they're supposed to work, if they're just slightly more active, what becomes more probable is that activation of neurons will get out of control. So neurons will spike more than they're supposed to. Neurons are too eager to spike. It's too easy to make them spike. And so spiking can become pathological. Instead of sending information from one neuron to another, there will be just waves of activation that don't serve any purpose anymore, that don't send information, but just sit there and, happen. And the body, the natural system that is supposed to regulate the level of activation, cannot catch up with that because the activation is so strong. It's called a seizure. And epilepsy is a condition of having seizures regularly. So that's, as you can see, one of the common consequences of having a mutation here. Kind of surprisingly for me, because I'm not a specialist, we don't see that many cases here, that many descriptions when the opposite seems to be true, when everything is not active enough. So I'm guessing that the reason for that is that if the channel is just slightly not good enough, you can have more channels. But if a channel is too good, it's pretty hard to have fewer channels, because if you have too few channels, just no spikes happen. So you have to have more, but then once you have more, but they are too eager to open, kind of the cat is out of the box, and you have a neuron that functions but is too eager to start spiking, which causes problems. But you can see that there are also other problems, and another word that's quite common here is pain. So there are classes of fibromyalgia that is also a kind of pain processing disorder, erythromelalgia, which is another type, and also insensitivity to pain. So that's the case when it's not too active but not active enough, and people have inactivations. So you can see that another typical thing that can happen, that there are several types of neurons, of sodium voltage-gated channels that seem to be in particular sitting in pain processing neurons. And because of that, if only they are affected, then there will be no seizures and all the central nervous system will work properly. But pain processing and communication of painful events to the body may be affected either in one end, in one direction or another. So either non-painful things will be perceived as painful, and then people have to sit with their hands and feet in the ice because even warmth is painful to them, or they may have insensitivity to pain, and then all the consequences with trauma that they cannot detect because they don't feel that their foot or their hand is damaged. Even though they feel touch, they don't feel damage, which is also very dangerous, but rare, but dangerous. All right. Yep. So See, everything, it's all like interesting and fun, but it's also deeply related to actual suffering of actual humans. And so neuroscience is, as all medical kind of biological fields, is a very involved field, because some of that is just genuinely curious and interesting on its own, but then there is this additional motivation that if you understand how it works, you can hopefully help people. And there are so many people on the planet that pretty much for any mutation, for any problem, if it's survivable, then there is probably a person with this condition. Maybe they don't know it. There is a thing called orphan diseases, when it's so rare that it's not even identified, but there is probably a person for any mutation that can be survived. There is probably a person with this mutation, and the consequences of it, if there are consequences. Okay, next. Oh yeah, how fast is action potential propagation? And I use this picture just because I really like it. It's not even relatively like relevant for that, but this is the entire nervous system of a human. So that's how you look like if you only look at the nervous system, huh? Okay, how fast is action potential propagation? If you touch something with your fingers, how quickly this information gets into your brain? The typical speed is from 10 centimeters per second to about 100 meters per second. So it's a huge range. Some neurons are slow. For example, pain, again, if your hand hurts, that's actually a good example. So if you hurt your hand, then there is sharp pain and then there is dull pain. And sharp pain is like, oh my God, I just touched a stove. So you really need to move your hand away quickly. And this is really fast. But then the other one that we mentioned is like, oh, poor little hand, I need to take care of it. So this part doesn't have to be fast. So the neurons that convey this slow, dull pain are very slow because nobody cares. They don't have to be fast, right? But everything related to motion probably needs to be fast. Everything related to sensation is more likely to need to be fast. What is it dependent upon? So why some neurons are fast, some neurons are slow, and how does propagation happen to begin with? I kind of mentioned already how it happens in really broad strokes. It's just action potential is happening here, and everything is heavily depolarized, so it slightly depolarizes stuff nearby. And because it depolarizes, the action potential starts happening here, so sodium starts to enter, and so in the next moment of time, you have action potential happening nearby. And it always goes in one direction, not in two directions. Why? Why doesn't action potential here cause an action potential like backwards and not just forward? What do you think? Why only one direction? Because you remember how action potential works? It goes up and then it goes down and then it recovers. And during this, like going down and recovery, it cannot be generated again, which means that this part is kind of tired in the sense that it's in the process of recovery, and so it cannot spike. So when this part spikes, it causes the naive part of the axon nearby spike, but not the part that spiked just before that, because it's not capable of generating an action potential for a few more milliseconds at least, right? Okay, how to make it faster? Are there ways to make it faster? Yes, there are two main ways. And one way to make just giant fibers. Why giant fibers allow faster propagation? It's something to do with electricity, with how currents work. And we won't go deeper into that because it's kind of a fancier topic where you need to actually write some equations to understand that. But one important thing that you kind of can relate to, okay, larger fibers, faster propagation, okay? And interestingly, many animals have these giant fibers. For example, a squid has a giant axon that goes through their entire body on both sides. So I guess technically there are two giant axons. What are they used for? If you were to make a guess, so you're a squid or you're like designing a squid. I will be playing this game a lot in this class. I usually imagine that you're Archangel Gabriel. So imagine that you're like creating the world and someone tells you, well, design a squid. And actually it's evolution that shaped it the way it's shaped, but we can try to reverse engineer it. Sometimes our guesses will be right, and sometimes our guesses will be hilariously wrong, and that's very important. And pretty soon we'll have some cases when if you try to reverse engineer something, you actually, like, it doesn't work, but in many cases it does. So let's try to make a guess, a sensible guess. If you know that an animal has giant fibers going through them, and action potential is really fast propagating in these fibers, why would these fibers be there? Why would they be beneficial for the animal? How does the animal use them? Use them. Make a guess. What behavior is it associated with? It's a horrible sound. I should stick to this. Okay. What? Is it feeding? Is it breeding? Probably not. Probably these are not the fastest, not the case when the speed of propagation is really important. So it's probably something like movement, but not any movement. What movement? Has to be so fast that, like, as fast as possible. Escape responses. If somebody tries to eat you, so if somebody tries to eat a squid and they like approach here and they like try to, that's somebody who tries to eat the squid, okay? The moment they touch the squid, the action potential will propagate through the fibers, and they're so large, and so I guess actually somebody may be on the other end. But anyways, so the moment when the squid needs to escape, that's another scary somebody, action potentials go through these fibers. I think they should go in the opposite direction. I think they should go like that. But the idea is that you can involve lots of muscles in this response really fast, and the squid can make this whatever escape maneuver that squids do. Some squids also have some ink that they inject in the water in order to hide, so lots of interesting stuff happening, and they make this fast movement and try to escape. And we will talk about escape maneuvers quite extensively for 2 weeks in this course, because escape maneuvers are really cool as a way to study how brains function, how nervous system functions. And one interesting comment is that a squid giant axon is not the same as a giant squid axon, because there are squid, but then there are also giant squid. And I recently learned from Twitter this year that giant squids actually don't have giant axons, only small squids have giant axons, which is funny and may not make sense at the first glance, but then you realize that, okay, if you're a really giant, then you're so slow that there is no benefit. Like a whale cannot, when you scare a whale, the whale probably doesn't jump. If you ever scale the whale, because a whale is probably what, you know? So there is no benefit in making communication super fast if you're really big, but if you're kind of medium size, and there is no benefit in improving communication if you're really small, because then something is fast, because then you're so short that everything is fast on its own. But if you're kind of medium size, when you still can respond quickly, but already takes a while for the information come from one side, from one part of your body to another, then there may be a benefit in optimizing the speed of propagation. Okay, and that's what squid do, and that's what insects do, and that's what invertebrates do. But we don't do that. We don't have giant axons, and yet whales also. But then communication within our bodies is actually pretty fast, and we use a different system. We use a thing called myelin, and I won't go too much in depth here, but I will just in really broad strokes mention that. So myelin is a special insulation, layers of essentially fat that's wrapped around our axons, and they're produced by different cells, not by the neuron, but by a different cell that in the brain is called oligodendrocyte, and in the body is called Schwann cell. So if you remember this horrible neuron that most people draw that has like a strange thing, and then it has like beads on them. So these beads are actually representations, like a mythical representation of these cells that insulate the fiber, the axon. And here's the microphotograph that looks like a vinyl record a little bit, where you can see that what happens is that when you have an axon, the cell really wraps around the axon and creates these layers of insulation by creating a spiral around the axon. So this vinyl record looking thing is actually like cell not cell, cell not cell, cell not cell, cell not cell, and so on. Okay? How does it help? In two ways. First, when everything is insulated, when your axons are insulated, when one of them is spiking, it doesn't affect the neighboring one, so you can pack them more closely. But another thing is that it makes everything faster, because so without insulation, when you have action potential here, the next moment it's nearby. When you have insulation, as it turns out, when you have an action potential here, at the next point, it triggers action potential on the side at the point between, like, between two cells, either oligodendrocytes or Schwann cells. So you can kind of skip this entire part. And that's the part that's a little bit tricky to understand if you don't involve like electric currents, if you don't try to describe the electric picture and how electric currents go in the system. So just believe me that that's the case. And then if you take an advanced course in cellular neuroscience, then you will learn it in much more detail. But what's interesting is that, so there are several things that are interesting about that, like three, I guess. First, this is our first encounter with a different type of cells that are important for the neural system that are not neurons, and they're called glia. And glia, here's the word like below here, glia are all the support cells. And it's interesting. So glia is very important. They're not neurons. They're also funny shaped, many of them, but not because they spike. They don't. And they don't actually collect information. At least that's not, they do, but that's not the main thing that they do. That's not the primary reason why they're shaped like that. They're shaped like that to help more neurons. So they reach to neurons in order to help them. And different types of glia help in different ways. So oligodendrocytes, they create this, they create myelination, while say astrocytes, they help synapses to function, and we'll talk about it later. So they feed neurons. Neurons are very lazy and arrogant. They don't want to make their own food. They don't. Some of them don't even make their own neurotransmitter. They really want it to all be served to them by glia, and so glia serves neurons. And there are lots of glial cells in the brain. In fact, apparently there are more glial cells in the brain than neurons. But yeah, back to the myelin though. Yeah, okay, this is the astrocyte that helps the synapses. This is the oligodendrocyte, and you know, all the stuff around the neuron, the axon. So the axon is running somewhere over there, and we don't see the axon, but we can see the oligodendrocyte, myelin, adding myelinations, different axons in this nerve. And what will happen? What happens if this if myelination is damaged in a human, or in parts where it doesn't work as well as it's supposed to? So one interesting part, apparently, where this is the case is the thing called photic sneeze reflex. When bright light makes people sneeze. So it doesn't happen in all people, but in some people it does. And it happens because we have this nerve in our head that is both sensory, so it brings information from the nose. No, what am I saying? Yeah, okay, right. Yeah, what I'm saying is true. There are nerves that bring information from the nose, but also in this same nerve are entirely different axons that control the. Is it true? I actually forgot. I should have checked that. I think there are axons there that control the shape of our pupil, that makes our pupil become narrower when we look at the bright light. That's my vague recollection. And so allegedly, although it's actually, my understanding again is that it's not quite proven, but allegedly the photic sneeze reflex, so people sneeze sometimes or find it easier to sneeze when they look at the bright light, happens because there is a little bit of short circuiting between these not quite myelinated axons. So axons that command your pupil to contract when you look at a bright light, by mistake sort of make neurons that come from your nose to the brain spike, as if there was something in your nose and then you sneeze. And not all people have it, but I think more than a half of all people on Earth have it. So slightly more than a half of all people actually have it to some extent. But a more classic example is multiple sclerosis, which is a sad disease, and it's a disease in which myelination, first it starts in the periphery, then happens in the brain as well, and myelination is affected, and myelination stops, like disappears. And the typical progression of this disorder is that people get worse, but then kind of get better, but then they kind of get worse again. And a lot of the intervention is actually about catching people when they're in remission and when they're not getting worse, and like preserving them at this state. And an interesting question is, so what happens when people get better? Because actually the myelin doesn't grow back. So it's not like it grows back. Unfortunately, it doesn't. But when it disappears, people lose the ability to walk, for example, but then they learn kind of to walk again. And one thing that we could do is to try to guess what happens. So there is a fiber, the fiber is myelinated, it works. Then later, one of the cells dies, so it's myelinated here, but here it's not. And now this fiber stops working, right? And the person cannot function. But then you give it a few weeks to months, weeks, I think, and they regain the ability to use this fiber. What happens? Make a guess. So what happens is that it switches from myelinated way of propagation to non-myelinated. So originally when the cell dies, there are no sodium channels here, because originally like this part was spiking, and then this part was spiking, and this part was bypassed. But then if this cell dies, sodium channels appear and populate this part of the axon. And it's slower and it's not as perfect. And in case of tightly packed fibers, it also means that they short circuit all the time. So the person doesn't recover completely, but they recover somewhat. And again, one of the major ways in which people think about this disease is, of course, how to prevent it, but also when it started, how to freeze everything in this point, because to some extent, our bodies are flexible enough that, we try to function, and we actually can function even if some myelination is gone. But when all of it is gone, unfortunately, the symptoms become worse and worse. Okay, so this is the end of the story, and see you next time.